Hello everyone, I hope you are enjoying the meeting so far. I would like to start by thanking Heiner and Pietro for the opportunity to present today. In this talk today, I will try to answer the question of whether we can stop Bulevirtide treatment through understanding, modeling the kinetics of HDV during therapy. The recent study by Tariq Asela and colleagues, published in New England Journal of Medicine, would be an excellent starting point, as they reported HDV negativity during and after end of treatment with bulevirtide and/or pegylated interferon. Under pegylated interferon alone, they reported that about 25% of patients reached HDV negativity by end of therapy and remained as such during follow-up. Though the study arm is small, the negativity rate or cure agrees with the literature. Under bulevirtide 10 mg daily alone, HDV negativity increases during treatment. However, during follow-up, HDV negativity, or let's call it cure, falls back to the rate achieved by week 48 on treatment. It is interesting to know whether early negativity is associated with cure. When bulevirtide 2 mg, keep in mind the dose, was combined with pegylated interferon, the results indicated that by week 96, 44% of patients achieved negativity. The negativity rate declined over time, ultimately reaching 26% by the end of follow-up. Notably, pegylated interferon plus 2 mg bulevirtide daily appear to achieve a higher rate of HDV negativity by the end of treatment compared to pegylated interferon alone, though both groups ultimately reach similar HDV negativity rates after the completion of therapy. Finally, when bulevirtide 10 mg, keep in mind the higher dose, was combined with pegylated interferon, the results indicate that by end of therapy, 70% of patients achieved negativity. And during follow-up, about 50% of the patients reached cure. Notably, the 46% HDV negativity with the PEG and 2 mg of bulevirtide appears, but not statistically powered, to be higher than with pegylated interferon alone. The extremely high HDV negativity by end of therapy that led to high HDV cure rates with bulevirtide 10 mg plus PEG suggests a welcome advance in HDV therapy. Historically, on-treatment hepatitis C levels served as an indicator of pegylated interferon-based treatment outcome. Given that finite bulevirtide-based therapy shows promise but is suboptimal, it's crucial to explore whether HDV kinetics during treatment can serve as a predictor of response. So mathematical modeling of viral kinetics during antiviral therapy could help to provide insights into drug mode of actions, to estimate drug efficacy, to understand HDV-HB surface antigen host like ALT interplay during infection and treatment, and to develop and optimize response-guided therapies. So what do we know about pegylated interferon mode of action? Using very frequent HDV kinetic measurements during pegylated interferon therapy, we previously showed that after a couple of days, during which HDV remained at baseline levels, two kinetic patterns were identified in responders. Here you can see a one viral kinetic pattern, which is called a flat partial response, in which a fast viral decline is followed by a plateau. The other viral kinetic pattern is called biphasic, in which you have a rapid decline followed by a slower decline in HDV RNA. We showed that the HBV, we found that the HBV surface antigen only declined in biphasic patients. So we developed a model that you see here on the right that accounts for HDV and HB surface antigen dynamics. This model is consists of target cells that are HBV surface antigen infected cells that could be infected with HDV and then become HDV productive cells, which could produce a new variants to the blood and also release surface antigen. Now, what we can do with this model, we could examine potential mode of action of interferon. We can block infection, we can block production of HDV, and then try to find out which of these mode of action can fit the data. And the model suggested that PEG interferon mode of action against HDV is mainly to block its production. So in summary, here we can show all the main findings of the mathematical modeling. One is to estimate that there is a delay of 9 days before HDV declined under pegylated interferon therapy. We also estimated that there is a high HDV burden of about 10 to the 9 virions which are produced and cleared in a patient per day. We also estimated that the efficacy of pegylated interferon to slow HDV production was about 96%. We also estimated for the first time the HDV half-life in the blood, which was about 3 days, and explained that the second phase of decline may reflect the HDV HB surface antigen cells death loss. So altogether, the potential clinical use of this model is that a flat second phase in HDV kinetics was associated with treatment failure, providing a potential early stopping rule during interferon treatment. So what do we know about Bulevirtide mode of action? Our modeling journey of Bulevirtide dynamics actually began when Pietro's group and colleagues published a frequent HDV kinetic study in 3 patients treated with Bulevirtide for up to 3 years. Historically, we know that the basic model predicts a monophasic viral decline under treatment that blocks virus infection. As shown here in the basic model on the top right that we used for pegylated interferon, Bulevirtide known mode of action in blocking HDV entry was included, and you can see that in green. The model fits very well the measured HDV viral decline in 2 patients, as you can see here, which exhibit a monophasic viral decline as expected during monotherapy Bulevirtide. Good. Now that we showed that the model explains the observed monophasic HDV decline by assuming that Bulevirtide blocks HDV entry, the next question would be, can we estimate Bulevirtide efficacy? Please note that the rate of the monophasic decline is driven by the rate loss of the infected cell loss or death, represented by the parameter delta, and the treatment efficacy in blocking infection. So to avoid this identifiability issue, one of the parameters, eta or delta, should be fixed. So we chose to fix eta to 100% based on its ability to block HDV spread in vitro and in vivo studies. While the basic model fits well to patients' data, assuming that bulevirtide only effect is to block HDV entry, it was not able to explain the transient HDV increase seen in one patient. Theoretically, the transient viral increase can be explained if one assumes that in addition to blocking viral infection, bulevirtide enhances viral production or it reduces viral clearance from circulation. So from model simulation, you can see that both mode of actions could fit well and explain this transient viral increase that was followed by the monophasic decline. So, however, bulevirtide is unlikely to enhance viral production, and we think that viral clearance by bulevirtide may be a more plausible mode of action if viral clearance from circulation is interrupted by bulevirtide blocking viral entry into hepatocytes. In summary, modeling suggests that bulevirtide may interfere with HDV clearance from circulation. I would like to highlight that in our recent uh, paper in eLife, uh, analyzing and modeling hepatitis C kinetics during liver transplantation, we showed that the liver in some patients not only produces HCV but also clears it from circulation, supporting the notion that blocking viral entry could reduce viral clearance by the liver in patient 1. A major challenge of bulevirtide-based treatment is its undefined length of therapy. The question is whether we can use mathematical modeling to predict the duration of therapy to achieve cure. We recently showed in a proof of concept clinical trial that modeling a hepatitis C kinetics in real time could identify patients for shorter duration of therapy to reach cure. The modeling concept is to use the measure kinetic data, you can see here in circles, before it go undetectable. This is marked by this yellow, filled yellow circle. Um, Before it go undetectable, to project with the model, you can see here in the curve, the time to reach the cure boundary, which we defined here as less than one virus copy in the total extracellular body fluid. This study showed that real-time response-guided therapy with DAA shortens treatment duration in about 40% of patients. And, um, and this is the first clinical trial in the field of viral dynamics using on-treatment real-time modeling-based response-guided therapy. Let's go back to the 3 patients treated with bulevirtide and applying the concept of using modeling to predict the duration of treatment to reach cure. Here you can see that modeling the patient number 1 and predicting the trajectory of the virus disappearing in the circulation, we can retrospectively explain why this patient had a viral rebound after 52 weeks because he did not reach the cure boundary. On the other hand, patient 2 was recently reported to reach cure 72 weeks after the completion of bulevirtide therapy. And here, with the retrospectively, we showed with modeling that he already um, reached the cure boundary after 50 weeks of therapy. So more research is needed to test and refine this modeling based RGT for Bulevirtide based treatment. Together with the Pietro's group in Milan, we are currently analyzing and modeling HDV kinetics of patients treated with Bulevirtide 2 milligram per day. As you may see, unlike the three patients previously discussed, not all patients have monophasic decline. Some patients have a biphasic, or in here, or a decline followed by a plateau. We have also non-responders and those that have a decline and have followed by a rebound. As you can see also, we characterize also the surface antigen kinetics in all patients which remain flat and analyzed ALT normalization during therapy. I would like to emphasize that the basic model cannot explain a non-monophasic viral decline under antiviral treatment that blocks virus infection. To illustrate that, we fit the basic model with a representative non-monophasic HDV decline case from our real-life cohort, and this is also shown in this meeting in poster number 30. As what you can see here, this patient have a biphasic decline with a rapid decline during the first 200 days, followed by a slower one, which the model cannot reproduce. And even if one can change the Bulevirtide efficacy in blocking infection or other parameters, it cannot reproduce or explain this biphasic decline. Nevertheless, the usefulness of the basic model for modeling HDV kinetics during a treatment provides important insights. And I will recommend you to, if you are interested, to read this recent review. To explain non-monophasic HDV decline and also account for surface antigen and ALT kinetics, we recently developed the following model. The model, as you can see, have these target cells as in the basic signs and infected cells. However, what we did here in this model, we assume that there are two types of short and long lifespan HDV infected cells. Here you can see also the ALT and the H, which account for the surface antigen. And for the model, as we did before, we account for Bulevirtide stopping entry, and potentially could have also other drugs like interferon, or lonafarnib, or nucleic acid polymerase, and others that could block other stages of the life cycle, like viral production, or blocking the surface antigen production. As you can see, the two population model fits well all three markers: the HDV RNA, the surface antigen remained flat, and the ALT declined to normal levels. The model explains the difference between the biphasic and the flat partial response decline patterns observed in the majority of patients. The first rapid phase of HDV decline that you can see here and here is explained by the HDV infected short-lived infected cells, which is I1 here in this cartoon, becoming HDV free. During the slower phase of decline, in the biphasic case, I1 disappeared, and then over time also I2 disappearing until the patient reached cure. Whereas in the flat partial case, the long-lived infected cells does not change. Our model can be further tested and refined by analyzing and modeling additional kinetic data, not only under Bulevirtide alone, is shown here, but also under Bulevirtide in combination with pegylated interferon, as recently published. In poster P30, in this meeting, we present our extended kinetic analysis showing that the majority of patients reached lower viral plateau under Bulevirtide monotherapy. We intend to continue to test and refine our model that was based on 48 weeks of Bulevirtide therapy with this longer data, which is up to 96 weeks of therapy. So in conclusion, ongoing efforts in analyzing and modeling HDV kinetics during Bulevirtide-based therapy are likely to develop and optimize response-guided therapy strategies for hepatitis delta. The emerging question may be not if we can stop Bulevirtide, but when. I would like to thank all my colleagues and funding agencies and all of you for your attention. Thank you. Bye-bye.